Reproductive cloning is basically creating a clone of a whole entire organism. We talked about molecular cloning, but that involved cloning just one gene or a few genes. But in this case, you're trying to clone an entire organism. And I'm sure you've heard about Dolly, the first organism that was cloned. So Dolly was created through reproductive cloning, and this image describes how reproductive cloning is done. So you need two different kind of organisms. One organism, this is this sheep, which is the nuclear donor. So this sheep donates the genetic material, and this is the organism that you want to clone. Then you also want a surrogate and a cytoplasmic donor. So the way it works is that you get eggs from this cytoplasmic donor, but you remove the DNA from it. So this is just the cytoplasm of an egg. Then the organism that you want to clone, you take memory cells from it, and then through a process of pulsing with electricity, these memory cells are fused with the egg cells. Then this cell that now has the cytoplasmic components of an egg and the DNA from the donor, then there are ways in the laboratory to induce them to start dividing and going under development to a stage which is called a blastocyst. It's an early stage embryo. So that early stage embryo is implanted into a surrogate animal from which the egg came from, and then that animal gives rise to the clone, which is Dolly. This is an exact clone of this organism. Now, this has been done, also cloning has been done with cats and so on, but these new cloned organisms, they tend to have a lot of health issues, and that's because we still don't know a lot about development of organisms and how their DNA changes. So in general, with the advent of molecular cloning and also sometimes cloning of certain genes into other organisms, we've been able to produce vaccines, antibiotics, and hormones at a reduced cost. Now, before we were able to clone the gene for human insulin into bacteria and produce it en masse and produce insulin en masse, it used to be very expensive to get insulin from anywhere. So a lot of people that had diabetes would pass away. But then the advent of molecular cloning reduced the cost of production of insulin. There's also a whole host of other therapeutic products that have been cloned in bacteria or in yeast or in mammalian cells at a very much cheaper price that you could otherwise prepare. So human insulin, human growth hormone, epidermal growth factor, interleukin-2, hepatitis B vaccine, erythropoietin, and factor VIII. All of these molecules are therapeutic proteins that treat a whole number of different conditions, and we have this available to us because of biotechnology.